So let's talk about how these enzymes are regulated. So isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase, both of these are upregulated in response to a decrease in the levels of ATP and NADH. So upregulated basically means that they're activated when there's a lower levels of ATP and NADH. Lower levels of ATP and NADH basically just means that high energy compounds are less, which means that the overall energy of the cell is less. And what we're basically doing is when we have less energy in a particular cell, we're making these two enzymes work faster. So the Krebs cycle, because these are the rate limiting steps, these are the enzymes that are going to control the rate of the entire Krebs cycle. When these are upregulated, they are working faster in the absence of ATP. The Krebs cycle is going on faster. It's working harder and faster, and because of that, more ATP is being produced, more NADH is being produced. ATP is available for direct use, whereas NADH goes to the ATC, reoxidized, just going to produce more ATP, and that's how the energy state of a cell can be improved. That's how we're producing more energy. If we were to look at this diagram, again, the same thing. We can see that isocitrate dehydrogenase is inhibited by NADH and ATP. What this means is that when you already have high energy in a cell, because these two compounds are indicative of a cell being in a high energy state. So when you already have quite a lot of energy in the cell, you want to inhibit this enzyme, and when you're inhibiting it, you're kind of slowing down Krebs cycle and thus producing less energy overall, because the Krebs cycle is now slower. Again, alpha-ketoglutarate dehydrogenase also inhibited by NADH, but also a buildup of succinyl-CoA. So basically, succinyl-CoA is the product of the reaction. When this is in abundance, when this is accumulating, that is also when the enzyme is going to stop working, because this again shows that you have plenty of energy in the cell. There's no need for more. And it's activated by ADP, which is going to tell you that there's less energy in the cell. And so you're activating the Krebs cycle and you're producing more and more energy, and that is exactly what the cell needs right now. So again, acetyl-CoA goes in. Acetyl-CoA goes in, and citrate is produced, oxaloacetate in the end, regenerated, cycle goes on. So yeah, carbon dioxide. This is basically just a summary of what I've already talked about, the products of the reaction. So I'm going to have you look at the purple box at the bottom. A single cycle is going to produce 2 molecules of CO2 excreted, 1 molecule of ATP, 1 FADH2, and 3 NADH. This is for 1 acetyl-CoA, 1 molecule of acetyl-CoA. We know that 1 acetyl-CoA comes from 1 pyruvate, right? And we also know that 1 glucose molecule gives us 2 pyruvate molecules. So what we're basically saying is that 1 glucose molecule is supposed to give you 2 molecules of acetyl-CoA, which means that this entire cycle is supposed to go on twice for 1 molecule of glucose. So 1 molecule of glucose gave us 2 molecules of pyruvate, and 2 molecules of pyruvate are going to give us 2 acetyl-CoAs. So 2 acetyl-CoA means 2 turns of the cycle, and 2 cycle just means that all of these products that we just talked about, they're going to get multiplied by 2. So 4 CO2, 2 ATP, 2 NADH, and 6 NADH. Just every product just gets doubled for one molecule of glucose. Again, this is the same thing that I just explained. 1 pyruvate molecule is required for 1 turn of the Krebs cycle, but since 1 glucose gives us 2 pyruvate, the Krebs cycle happens twice for every glucose molecule. So all the products that we just talked about get doubled for 1 glucose molecule. Okay, so we're just to sum everything up, let's talk about the overall number of ATP that is produced throughout the entire process of aerobic respiration. So just some ground rules. One NADH molecule, after it goes through the electron transport chain, is going to give us 3 molecules of ATP, and one FADH molecule, again after having gone through the ETC, is going to give us 2 ATP molecules. So think of it as a little key. So yeah, glucose, let's talk about glucose first. It says here cytosol, the rest of everything happens in the mitochondria. Okay, so glucose is going to give you 2 ATP just right off the bat. Net amount, again we talked about the 4 minus 2, the net amount is 2. And glucose is also going to give us 2 NADH. 2 NADH means 6 ATP. You get 2 NADH from PDH as well, from the working of PDH. One pyruvate is converted to acetyl-CoA, so that's another 6 ATP. One pyruvate to one acetyl-CoA is just 1 NADH, but since this happens twice, because one glucose is going to give you 2 pyruvate and 2 acetyl-CoA, so overall 2 NADH as well. The Krebs cycle, 2 ATP, 6 NADH, 2, and again, the Krebs cycle has been multiplied by twice because one Krebs cycle is going to give you 3, not 6. But since we've already multiplied it by 2, so 6 NADH, 2 FADH, 2, 4 and 6 3s are 18. And if you were to add this all up, you would get 38. And that's the net amount of ATP that one molecule of glucose yields through the entire process of glycolysis first, PDH second, the Krebs cycle, and after the Krebs cycle, the electron transport chain. All of these together, the entire process of aerobic respiration, gives you 38 molecules of ATP. And that's it. Thank you so much for tuning in. Hope you understood the lecture well. Just for reference purposes, the resources that I used here were the biochemistry book, which is Lippincott Illustrated Reviews, the 7th edition in particular. And thank you so much for listening. See you in another video soon, inshallah. Bye bye.